Good day, everyone. We have reached the final chapter, Chapter 9, Heavy Metals Pollution. Unlike other pollutants, which may visibly build up in the environment, trace metals in the environment may accumulate unnoticed, to toxic levels. Heavy metals are commonly defined as those with a specific density of more than 5 grams per centimeter cube. Heavy metals are serious pollutants in our natural environment due to their toxicity, persistence, and bioaccumulation problems. Heavy metals, in aquatic systems, can be from natural or anthropogenic sources. Currently, the anthropogenic inputs of metals exceed natural inputs. Organisms require trace amounts of some heavy metals, including cobalt, copper, iron, manganese, and zinc. However, the excess metal levels in aquatic may pose a health risk to humans and the environment. Excessive levels of essential metals, however, can be detrimental to the organism. Non-essential heavy metals, of particular concern to surface water systems, are cadmium, chromium, mercury, lead, and arsenic. Heavy metals can be divided into 4 major groups, according to their pollution potential. Very high pollution potential such as arsenic, cadmium, chromium, copper, mercury, and lead. High pollution potential, such as barium, manganese, iron, and uranium. Moderate pollution potential, such as bromium, kalium, lithium, and nickel. Low pollution potential, such as gallium, magnesium, and tantalum. Metals pollution has drawn increasing attention worldwide due to a dramatic increase of anthropogenic contaminants to the ecosystems through the air, water, and soils. Pollution of the natural environment by metals is a serious problem because these chemical elements are indestructible. Most of them have toxic effects on living organisms when they exceed a certain concentration. Some metals are highly toxic and persistent, and have a strong tendency to become concentrated in marine food webs. There are 2 main processes the metals can enter an organism. Bioaccumulation, biomagnification. Do you know how to differentiate it? The accumulation of heavy metals in superficial sediments, from industrial effluents, and urban sewage discharged into the aquatic environment, without proper cleaning will easily be identified through metal spatial variations in sediments. Geochemical mapping can be used as a tool for visualization, which is enhanced by computer-aided modeling using geographical information system, to make it easier to identify the possible locations of the contaminated area. Heavy metal concentrations in harbor or estuarine sediments usually are quite high, due to significant anthropogenic contaminants loading carried by the upstream of tributary rivers. Therefore, sediment analysis is a sound tool for the assessment of marine pollution, and they are widely used as geomarkers for monitoring and identifying the possible sources of pollution. Sediment can preserve a continuous record of regional and even global environmental changes. To some extent, sediment is the mirror of sedimentary environmental changes, which can reflect the biological, geodynamic, and geochemical process of former conditions. Lying in the second trophic level in the aquatic ecosystem, shellfish species have long been known to accumulate both essential and non-essential metals. Many researchers around the world have reported the potentiality of using mollusks, especially mussel and oyster species, as bioindicators or biomarkers for monitoring the metals contamination of the aquatic system. Besides serving as a biomarker for marine pollution studies, mollusk species have also been used in ecotoxicology and toxicity studies. These figures show some example of the mollusk species that commonly use as biomarker or biomonitor. When we study the pollutant in the organism, the safety of food to the consumer is a major concern, to ensure the safety of consumers. Therefore, metals analyses of the aquatic organisms can provide important information on the degree of environmental contamination and potential impact of seafood, especially fish and shellfish consumption. What are the sources of heavy metals in our environment? Heavy metals of natural or anthropogenic origin is transported by rivers and transferred to the coastal marine system through the estuaries. These pollutants can enter the coastal environments and oceans by two means. The first one is natural processes. For example, erosion of ore-bearing rocks, wind-blown dust, volcanic activity, and forest fires. The second one is from human activities, such as atmospheric deposition, rivers, and direct discharges or dumping, wastewater. Marine aquatic organisms can accumulate heavy metals from various sources in their surrounding environment. Once the heavy metals enter the food chain, they may accumulate to dangerous levels and be harmful to human health when consumed the polluted organisms. Heavy metals accumulation depend on several factors, such as environmental concentrations of metals in water and sediments, species of organisms, body size, and age of the marine organisms. Different concentrations of metals can also be found in different organs such as stomach, gill, muscle, liver, in the same biological sample. For example, in fish sample. Besides that, the entry may be a result of direct discharges into both freshwater and marine ecosystems, or through indirect routes, such as dry and wet deposition, and land runoff. Important natural sources are volcanic activity, continental weathering, and forest fires. How can marine sediment act as a marker for pollution studies? Sediment are widely used as geomarkers for monitoring and identifying the possible sources of pollution in the coastal environments, since sediments are the main sink for various pollutants. Most metals are bound in the fine-grained fraction, or less than 63 micron-meter, mostly because of its high surface area to grain size ratio and humic substance content, where they have a potentially greater biological availability than those in the larger sediment fraction. From size of 63 micron-meter up to 2 mm. Sediment cores can provide chronologies of contaminant concentrations and a record of the changes in concentration of chemical indicators in the environment. The metal accumulation rates in sediment cores can reflect variations in metal inputs in a given system over long periods of time. Next, I will explain about biomonitor of heavy metals. Individual biomonitors respond differently to different sources of chemical elements, for example, in solution, in sediments, or in foods that they eaten. Therefore, we have to use a correct biomonitor that can reflect the element bioavailability in all available sources. Ideally, species to be chosen as biomonitors for monitor the environment should fulfill several criteria such as should be sedentary, easy to identify, abundant, long-lived, available for sampling throughout the year, large enough to provide sufficient tissue. Living organisms can transport pollutants and contaminants into, within, and out of the marine aquatic ecosystem. These organisms can ingest the pollutants via water and food, and inhale them as they breathe and feeding. Once pollutants in the body, some contaminants pass quickly, while others can be retained for long periods and accumulate in body tissues. The choice of a suitable biomonitor needs to consider the potential sources of metals to the biota. For example, seagrass, not in contact with sediments, will take up heavy metals from dissolved sources only. Suspension feeders such as shellfish organisms take up metals both directly from seawater and from the suspended particles collected during feeding. So, what are the effects metals on human health? This figure shows how the contaminants in sediment move up to the food chain. The contaminant that enters the aquatic environment will settle down to the sediment. Then the shellfish organisms will uptake the contaminants through sediments and particulate. Fish will eat the shellfish, so the contaminant will transfer to the fish. The predator species, bigger fish, will uptake the contaminant from the prey. Lastly, the contaminant will be uptaken by humans when we consume the fish. Generally, human health problems associated with trace metal contamination have been well highlighted in the literature. In spite of the relatively low level of industrial activity in less developed regions, there is nevertheless a high potential of toxic heavy metal pollution. Bioaccumulation, another way in which pollution can harm the environment, is the internal accumulation of a pollutant that is very difficult to degrade or expel from the body. Every organism accumulates pollutants during its lifetime, and if it is eaten, the pollutant are transferred to the predator or to higher trophic. Hence, bioaccumulation especially targets the top predators of the food chain, including humans. Do you notice that humans are the top predator in all environment? In marine, in the land, in air. This figure also shows the flow how the contaminants uptake by organisms through primary organisms. In this case, algae is the primary organisms, and they uptake to contaminants through the water column. And lastly, pass to humans if we consume the organisms from the ocean. Let's look for one example. Oysters can be swallowed or masticated normally, increasing the surface of contact between food and digestive fluids. The consumer will consume the whole soft part of the oyster. Therefore, metals content is examined in toto or oyster flesh. One of the most dangerous metals that occur in our environment. Mercury. Mercury is very low level in the environment, less than 0.1 ppm. The permissible level in seafood normally not more than 0.4 ppm. The concern of mercury pollution starts from the incident of the Minamata Bay, Japan. The disease was first detected in the year 1956, but the mercury emissions continued until the year 1968. In Minamata, Japan, inorganic mercury was used in the industrial production of acetaldehyde and discharged into the nearby bay as wastewater, and was ingested by organisms in the bay. Fish and other organisms in the sea were contaminated, and eventually, residents who consumed the fish suffered from methylmercury intoxication, later known as the Minamata disease. But even after the emission of mercury stopped, the bottom sediment of the polluted water contained high levels of this mercury. So till now, the impact is still visible. That's all for heavy metals pollution chapter. Thank you.